Hello folks, and welcome to a video lecture on enzymes. Most enzymes are proteins, but recently some enzymes were discovered to be made up of RNA. We call these ribozymes, and they function to speed up the production of RNA for use as genetic material. Let me tell you first what enzymes are, how they function, and how the cell can control or regulate how they work inside the cell. You can see the secondary structure of the alpha helices and the beta sheets, the tertiary structure of the entire protein folding in on itself, and then the combination of tertiary structures to make the functional quaternary form. Here's another model of an enzyme. This one happens to be of an enzyme called catalase. The chemical it works on is generally called a substrate. In this case, the substrate is hydrogen peroxide, H2O2. We can illustrate how enzymes work best by using simplified illustrations like this one. So what are enzymes anyway? Well, they're catalysts in biological reactions. That is, they speed up reactions. Here's a graph that shows the energy of a reaction. At this point, we probably have looked at energy of reaction graphs like this already. This one happens to show an exergonic and therefore spontaneous reaction. This reaction will proceed forward spontaneously, but because the reactants have an activation energy, it could take months for this reaction to run its course. The reactants can slowly absorb the free energy of the environment to activate it. However, introducing an enzyme can greatly reduce the activation energy and therefore increase the rate of the reaction immensely. In most cases, one enzyme can change on the order of thousands of substrates per second. Here's a look at a model of how the enzyme works on its substrate. The substrate, or reactant, is in one form here. The enzyme has an active site for that substrate. The two come together. This lowers the activation energy of the reactant, and the reaction proceeds. Notice, only the substrate has been changed here. Enzymes are never changed by the interaction, so they can perform their duty over and over again. Now here's an interesting point. In many cases, the enzyme that works to move this reaction in this direction is the same enzyme that runs a reaction in reverse. In many cases, as the products start to build up, the enzyme can run the reaction in reverse until equilibrium is reached. This also dispels the misconception that enzymes are only used to break down molecules. On the contrary, their only job is to lower the activation energy. They don't do the actual work of breaking down or building up. Free energy does that. To run a reaction in reverse, in this case an endergonic reaction, there's still an activation energy. The free energy to drive this reaction is usually from what we call energy coupling, which I discuss in the video titled Energy Transformations. Now you may have noticed that the active site had a shape that fit the substrate. Enzymes have a specific conformation or shape. The active site of the particular enzyme has to fit the shape of the particular substrate or it won't work. This tells us that there are about as many enzymes in cells as there are chemical reactions. That's thousands of enzymes. Not all of them have been discovered yet. You could be the next to discover an enzyme that could help us understand a disease or even find a cure. This enzyme-substrate specificity is sometimes called the lock and key hypothesis, whereby only one key fits the lock. Any other key simply won't work. We also think that as an enzyme and the substrate come together, they both go through a conformational change or a shape change. This is an interesting notion called induced fit. The enzyme returns to its normal conformation when the substrate lets go. Now, some enzymes need another compound to make them work properly. Chemicals called cofactors help make an enzyme active by acting as an intermediate at the active site. You know some of these cofactors. Some of them are minerals we need in our diets. Others are organic compounds, in which case we call them coenzymes or vitamins. Yes, that's why vitamins are important. They're needed to make certain enzymes work. There are generally 4 ways enzymes work to lower activation energy. These are in no particular order. One way is that they help by orienting the two or more reactants so that they react more easily, sort of introducing them to one another. Another way is they stretch or bend the substrate molecule and weaken its bonds, making it react more easily. Another way is that they can alter the environment of the substrate, such as lowering the pH. And in some cases, it's known that the active site can actually take part in the reaction. Subsequent steps are taken to restore the R group that took place in the reaction to its original state. So ultimately, the enzyme remains unchanged after the reaction. Enzymes like to work under certain environmental conditions. They're said to work optimally, that is, best under these conditions. But not all enzymes are alike, and they've evolved to work under specific conditions such as pH or temperature, or the combination of the two conditions, as shown by this graph, showing 3 different enzymes and their optimal conditions. This is a case of 2 enzymes in our digestive system. You might know that our stomach environment is very acidic. Pepsin is an enzyme that works best at low pH. Trypsin is an enzyme that works best at high pH, and that works in the small intestine. Other enzymes might have optimum temperatures at which they work. In the lab, you may be asked to think of a particular environmental condition and design a test to see its effect on the enzyme's activity. There are more conditions. Now, the environment can affect an enzyme's conformity by altering its shape, usually its tertiary structure. This is a denatured enzyme that simply won't work in this shape. Enzymes can also be altered by their chemical environment. Some chemicals mimic the shape of the substrate that the enzyme is supposed to work on. These mimics compete for the active site. This is called competitive inhibition. This kind of inhibition, however, can be overcome by simply adding more substrate. Competitive inhibitors, however, can also have dangerous side effects and are so-called poisons. Ricin is a competitive inhibitor that blocks enzymes necessary for protein production. Ricin has been used as a chemical weapon since World War I. It's produced by a plant called the castor oil plant. Ricinus communis is its scientific name. It creates a toxin in its bean seeds as a way to keep animals from eating them. Since it disrupts the protein production, the poisoning can linger for days before a person dies from the poisoning. A few grains the size of salt crystals is enough to kill an adult human. Unfortunately, it's still used as a chemical weapon, not only by the plant, but by humans. Other types of inhibitors don't block up the site, but instead bind to another site on the enzyme. The binding of this additional molecule changes the conformity of the enzyme and its active site, rendering the active site inactive. This is called non-competitive inhibition. The cell itself takes part in inhibiting its own enzymes. The cells don't always need enzymes to be doing their work, so they've evolved a way to turn the enzymes off or inhibit them from working, and a way to turn them on or activate them when they do need them. If a cell needs to stop an enzyme from working, it can create a compound that works like the inhibitor in the previous example. It binds to a site other than the active site and then ends up warping the active site out of shape. When this takes place in cells, it's called allosteric inhibition. The term allosteric can be dissected into its Greek roots, allos meaning other, and steric meaning arrangement or shape. This process can also work to activate an otherwise inactive enzyme. In this case, it would be called an allosteric activator. In some metabolic pathways involving several enzymes, the products at the end of the pathway can act as an allosteric inhibitor to the enzyme at the beginning of the series of reactions. Subsequently, as the product builds up, the cell may not need more of it, so the product inhibits the primary enzyme. As the concentration of the product lowers, the enzyme is freed up and becomes active again. It's a self-regulating factory, like a shoe factory that has a surplus of shoes. If the shoe sales slow down, then the factory should slow down shoe production. This is simply supply and demand at the molecular level. That's enough about enzymes for now, but you should know that the study of enzymes is so important that scientists are asking for help from everyday people like you. They've created a game you can play that could actually help them make new discoveries about enzymes. It's called Fold It, and the link to it is here. I also have a link to a short video about it on our webpage under Cell Metabolism, the same page this video is listed on. You should at least watch the video. That's it for a lecture on proteins. I hope you learned something, taken some notes, and if you have any questions, watch the video again or bring them to class. Until then, be well.
Patty cake, baker's man. Bake me a cake as fast as you can.
Dude, that's not
Hey,
roll it.
no,
That's
why'd
not
you,
roll it.
You dropped it,
You,
man.
that's not roll it.
You quit.
That's not roll it.
No, yes, it is.
No, it's
Are
not.
You,
No, you don't. I'm not going to do it if you don't know.
I know how to roll it. Are you?
Stop.
Hold on.
You don't even. Who's that?
Look, Creeper.
Hello, creeper.
What's good? What's good?
All right, but
Okay,
you don't know how to roll
patty
it.
cake. Let's go. Come on.
Are you-
Yeah, I know.
are
I
you
know how to
gonna
roll.
roll it?
I invented it.
Are you going to roll it?
I started it.
All right.
Yes.
It doesn't start like that.
It
doesn't.
We're
it
starting
doesn't-
over. We're
stop,
starting over.
Stop.
Okay, come on.
Stop.
Just start over.
It
Let's
starts
go.
with this bump.
Easy.
Ready? Ready? Patty cake, baker's man. Bake me a cake as fast as you can. Roll.
Ow!
Oh,
Hey,
dude,
dude,
what's that? What's that?
You
You
bit me.
stopped
You're
again.
saying
You
scratched
stopped
me. You got
again.
some talents,
Little
man.
baby.
Hey, don't call me a baby.
Little
Don't.
kitten.
Hey, don't call me a baby. You remember last week what happened when you called me a
Get
baby?
it close up.
Stop.
Dude, get it close
stop.
up.
Hey, get that camera away
Oh,
from me.
little
Stop. Hey,
baby.
that hurts.
Little
Stop.
baby.
Stop it. Don't call me a baby. Yeah.
Oh, dude, what was that? Stop. Stop.
It's payback,
Stop.
baby.
Stop.
Payback.
Just
you
stop
little
right
baby.
now.
Come on, Nate, no.
Stop right
Come
now.
on.
We
Patty
don't
cake.
hit.
Let's go.
We
Patty
oh, dude, I'm not gonna do it
All
if you hit me again.
Okay.
Just stop.
Patty cake then.
Just
Patty
stop.
cake.
No.
Patty cake.
One
Come
more
on,
time?
Let's go.
All right.
Come on.
Ready?
Come on, let's go.
You ready?
Patty cake, baker's man. Bake me a cake as... Hey, you quit. You're stopped.
Whoa!
I'm tired of you quitting like that.
I'm done.
Do not quit. No more quitting. Patty cake. Patty cake. Come on.
It's over.
No, it's not over. It's never over.
Shut this camera off right now.
No.
Shut the camera off. I'm done. I'm out of here.